Hello everyone, my name is Ayumi Tsuyuki. I'm from Duke University and I'm so excited to be here today to talk with you all about the analyses of hypoxia and metabolism that we did. I want everyone in the room to take a deep breath, hold it, and see if you can hold it for the rest of my presentation. Probably not, but I hope this highlights to you just how important oxygen availability is for us as a species. Quick trigger warning before we get into some scary stuff. You probably are way too familiar with this graphic here, but I wanted to put it up here to highlight the 4 metabolic pathways that we studied, ranging from glycolysis to fatty acid metabolism, oxidative phosphorylation, and reactive oxygen species. And our goal was to determine how all of these pathways interact with hypoxia. Oxygen availability has been crucial for evolution, starting with the Great Oxidation Event, roughly 2.5 billion years ago. The rise in atmospheric oxygen levels has created this huge diversity and complexity in evolution. And along with those changes in evolution, we saw both phenotypic and genomic differences and changes to gene networks. So we hypothesized that the early eukaryotes had very tightly connected metabolic and hypoxia pathways, and then later on, gene duplication and genome expansion further tuned those pathways. A few quick notes about the analysis we're doing. When you have a stimulant, such as coffee, that's going to induce different changes in expression for different species and different genes. And that in turn will sort of affect gene connectivity, and gene connectivity is important for us to study because it gives a sense of gene essentiality and sort of insights into how different genes are interacting together. And all of that will induce variations in the phenotype. So for our methods, we pulled 5 data sets, hypoxia and our 4 metabolic pathways from Molecular Signatures database. And those were the hallmark human gene data sets. So we ran those through Ensemble and gProfiler in order to find the orthologous genes that share the same function in 14 other species. After that, with these gene data sets, we ran them through Cytoscape and R to perform our analysis. So here you can see sort of what we expect, and I want to point out these are the species that we looked at, ranging from these sort of more basal yeast and invertebrates down to fish, mammals, and human. And what we were looking at here was just looking at the presence or absence of the orthologous genes in the hypoxia pathway. And you can see that these more basal organisms tended to have fewer hypoxia genes as compared with the later species. And we also wanted to sort of verify that this was correlated with genome size and evolutionary order. So those represent our most basal invertebrate organisms that have smaller genome sizes and fewer hypoxia genes. Next, we turn to look at connectivity. So here you can see in this hypoxia gene network, these dark blue genes represent the genes that were conserved in yeast and all of our other species. And you can see that they sort of look like they're more tightly embedded in the network than, say, the genes over here that only pop up in the human species. So we quantified that, and just to reiterate, we expect that the smaller networks have more tightly connected genes, and then as the network expands, the connectivity will be lost. And so this heat map is showing the change in connectivity for each gene in each species as compared with this early C. intestinalis. And the blue sort of signifies what we expect to see, that there's this loss in connectivity across the evolutionary scales. We also did this for our other metabolic pathways, and we found glycolysis. Once again, you can see that preponderance of blue right there that signifies this loss in connectivity. And we found that glycolysis overall showed the most significant changes in connectivity, which is especially noticeable when you contrast it against oxidative phosphorylation, which shows very little changes. And we believe that oxidative phosphorylation, we're thinking that maybe it's because it's so essential, that's why there wasn't much of a change in connectivity. We also wanted to see how the pathways interacted. So we looked at the proportion of bridge genes, which were the genes that were shared between hypoxia and each of the metabolic pathways. So you can see here, once again, so glycolysis appears to be the most tightly connected with hypoxia due to that higher proportion. And overall, for all of the pathways, there's this sort of rough decreasing trend, which sort of correlates with what we saw earlier in terms of loss of connectivity. Once we had identified glycolysis as this pathway of interest, we wanted to narrow the focus a bit and study it in marine mammals, which provide a naturally evolved system that enables us to study how oxygen availability impacts organisms. So we obtained this skin biopsy here from researchers at the Duke Marine Lab and placed them in a cell culture plate. After about a couple of days, you can see that the cells come, this is a zoomed in view of the biopsy, so the cells come from the biopsy and start attaching to the plate. And then after about a weekish, the cells proliferate. So we cultured our cells, and then we applied conditions of either normal or low oxygen. Hypoxia in this case was about 1% oxygen. And then we measured the mRNA expression with RNA-seq. And so that allows us to compare the expression levels across and the cellular response across different genes and different conditions. So we isolated the glycolysis genes and we compared them with human, and you can see this sort of general sort of upregulation. And so the response is really interesting. When you compare it with all the marine mammals that we studied, you see a very different trend in terms of the expression. Particularly, you can see this sort of like some of the genes are downregulated where humans are upregulated. And so overall, this shows a sort of tempering of the glycolysis response that reflects some of what we saw in our computational analyses. And so we wanted to see, okay, why do we see this tempered response? And one of the mechanisms that we hypothesized could have something to do with it was an increase in spare respiratory capacity. So what we have here is we measured the oxygen consumption rate using a seahorse assay for all of these different species. And you can see that goose-beaked whale, which is the organism that dives the deepest, has the highest oxygen consumption rate. And then it's sort of followed in gradations down roughly by diving ability. What's really interesting, though, is when you look at 1% oxygen, when the oxygen has dropped way down low, the goose-beaked whale is actually able to roughly maintain the same oxygen consumption rate when all the other organisms have dropped down way low. And so this was a really interesting result to us. And so just a few quick notes about what I mean by spare respiratory capacity. Generally, you're going to have a sort of basal respiratory capacity here. And then when your cells are put under stress, then you're going to want to increase your respiratory capacity and increase metabolism up to a sort of maximum. But if you have low spare respiratory capacity, that maximum is not going to be that much higher. And so your cells have a poor adaptation to stress, which often results in distorted mitochondria. On the other hand, if you have high spare respiratory capacity, like what we think we see in the marine mammals, then you have high adaptation to stress, and it means that your respiratory capacity can exceed way past your basal amount. And that often is correlated with healthier mitochondria. So all of this led us to wonder, what does the mitochondria look like in the marine cells? So we did some stains. You can see the mitochondria in the human here. And what's really striking is when you look at the goose-beaked whale. And so for a bunch of the cells we tested, all of them showed this huge increase in the number of mitochondria as compared with the human. And so seeing that there's this increase in mitochondrial volume, we probed a little bit further, and we asked Well, is this increase in mitochondrial volume linked to mitochondrial biogenesis? So one gene that kept popping up in our analyses was PGC1 alpha. And PGC1 alpha is a well-known regulator for mitochondrial biogenesis. It's known to remodel muscle tissue to become less glycolytic and more oxidative in nature, which kind of reflects the stuff we saw before with the glycolysis decoupling. And it's also involved with some of the reactive oxygen species, fatty acid metabolism, those pathways we were studying earlier. So we identified this gene, and we performed RT-qPCR in order to measure the expression values and compare with human. And as you can see here, there seems to be this trend of an upregulation of PGC1 alpha, which is in contrast with the downregulation in humans, which brings us all the way back. So we think that that the increase in mitochondrial volume that we saw could be linked to an enhancement of mitochondrial biogenesis thanks to genes like PGC1 alpha. So just to sum this all back up again, we saw that as the genome size changed and the evolutionary sort of order changed, the number of genes in each pathway was correlated with that. We saw that invertebrate genes tended to be more tightly connected than the vertebrate genes. And we identified glycolysis as a pathway of interest in terms of the significance of their changes in network connectivity and its involvement with hypoxia as compared with our other metabolic pathways. When we looked at the marine mammals, we saw a sort of decoupling of glycolysis and an increase of spare respiratory capacity, which we believe is linked to an increase in mitochondrial biogenesis due to genes like PGC1 alpha. In the future, some of the things we're looking at, we'd like to look at a phylostratigraphy analysis of the genes that had the highest changes in connectivity, because this could help us sort of infer the gene's evolutionary origins. We also would like to identify some sequences that would be responsible for the changes in connectivity that we saw across different species and different pathways. And finally, we aim to look more deeply into the role of PGC1 alpha in human cells. And potentially, the sort of dream would be if we can get the human cells to match the response in marine cells, maybe we all could dive the same way whales do. So stay tuned, that's undergoing. But I want to thank everyone who helped us out with these analyses and this work, and thank you to the organizers of this conference. Thank you. Yeah.
Great, very, very interesting. I'm interested in what might go on with cancer cells in humans. Would you think that they revert back to some evolutionary ancestral state, or is it something completely different?
That's a great question. So my lab is involved with looking at sort of hypoxia and cancer, which makes a lot of sense. I guess, so we actually are looking at, right now, we're sort of in the middle of analyses looking at how the results that we saw from these early invertebrates, like yeast, can be applied to human tissues and cancer. So I can't say for sure what's going on there, but we did see that cells like blood, muscle, and testis sort of have a similar response in a lot of ways to the early yeast invertebrate. So yeah, I think there's a lot to be done with that work, but there is a possibility that some of the stuff we saw could be applied with the early response, the early invertebrates, and cancer. Yes?
It's very interesting. What do you think the adaptive significance is? Because you're consuming so much more oxygen than the whales, but
Okay, please, that time or? Oh, okay. I can talk to you about this. Cool. We're switching from whales to butterflies now. Hi, everyone. Thanks for coming. I'm Kathy. I'm a PhD candidate at Stockholm University, and I'm working with host plant adaptations in polyphagous butterfly larvae. And just to make sure we're all on the same page, I need to click here. Insects can have very strong associations with the host plants. They use them as a shelter for reproduction or as a food resource at different developmental stages. And depending on how many different plants an insect use in this context, we can distinguish between the specialists, so those that use one family or one species. And on the other hand, we have the generalists, so those that can use a broader variety of different host plants, different families, sometimes even different orders. And what we are doing is that we're trying to understand how a generalist butterfly can feed on host plants that are sometimes very diverse in their morphological, physiological, and chemical properties. So what are the mechanisms, what adaptations are required? And one thing we already know is that larvae that are feeding, or that were fed on different host plants show very different gene expression profiles. And this is what you see here in these spherical PCA plots. Every gene is one dot, and then they are joined together based on expression similarity. And to make this clearer, you also have a color gradient. And as I said, you see that on the different host plants, the gene profiles, the expression profiles look very different, especially when you compare larvae that were reared on Urtica versus the one that were reared on Rebus. It looks quite of the opposite. So we have host-specific transcriptional response. And this is not only true on this Polygoniaceae album, but also in other nymphalid species, which is summarized in this pool here. To look a bit close into that, you see that the reaction norm, gene expression response, for all these nymphalid species on the different host plants look more or less the same. And the cool thing about this is that this is even true for Polygonia faunus and Nymphalis xanthomelas that are not using Urtica as a host in nature anymore. Now, based on that, host plant adaptations can be understood as modules, sets of co-expressed genes and the resulting phenotypes. These modules seem to be conserved, and they're also maintained even though a host is not part of the realized repertoire anymore. And given that we see some, opposite gene expression patterns, we think that this could also be an indicator for potential physiological trade-offs. One thing that was not very clear is, can the larvae switch between these different modules? And if so, how quickly can they do this? And to test this, we again used Polygoniaceae album because, best butterfly in the world. And it feeds on different species from 4 different orders of plants, so something very peculiar. We did some host switch experiments where we reared and moved larvae between 3 different host plants, Urtica dioica, Salix caprea, and Rebus crispa. So once a tiny larvae hatched, we put it on one of these 3 host plants, in this case Urtica. We let it grow, and then once they reached one larvae reached the 4th insta, we moved it to a new host plant. Could be the same species, like a control, or it could be one of the alternative hosts. And to see whether this switch had an effect on the transcriptional response, we collected gut tissue 2 and 17 hours after the switch and did some RNA-seq on that. We did this for all the different treatments and host combinations. So we had 9 treatments in total, and for each treatment, 2 different sampling points. And the first thing we wanted to see is, does one of the hosts have a major effect on the gene expression response? And here the answer is that it depends a little bit on the time point, because after 2 hours, we see that the gene expression pattern, or the highest number of differentially expressed genes, is very much explained by the first host, while after 17 hours, this moved, and the highest number is found in association with the second host plant. And then we wanted to see what is happening when we compare plants that were larvae that were switched between particular plants. And for this, we compared larvae that were switched with those that were exclusively reared on the same host plant, so a control. And this is what we tried to summarize here. In upset plots, so you have the comparison groups here, so a larva that was moved from Rebus to Urtica, the corresponding control line, and you see
the total number of differentially expressed genes per group, and also how many of these genes are unique to this contrast or shared between different groups. Going back to the overall data, or not? Yes. So this is a lot. I don't want you to look at all the details. The take-home message here is that groups that have the highest number of differentially expressed genes were not the same between 2 and 17 hours. And after 70 hours, the highest number of differentially expressed genes was associated with switches that involved rebus. Good. Now we had a quantitative idea of what is different. We also wanted to know what do these differences actually mean. And so we compared the patterns or the differences to our expectations. What does that mean? We basically wanted to see if a larva is moved from, for instance, urtica to rebus. We first compared the two control lines, those exclusively reared on the same host plants. We compared the gene expression pattern and said every difference that we see here is also what we can expect to see after the switch. And then we looked at the gene expression pattern of a larva that was switched and checked, do we have a match with what we see between the controls or not? And when they matched, great. This would be an indication for adjustment to the second host. But it could also be that there were only differences between the controls, but not after the switch, which could be indicative for its gene expression that still corresponds to the first host. In the opposite way, it could be that we see differences after the switch that are not present in the controls, or they're actually opposite to what we see in the control lines, something that we summarize as a stress response to the switch. Good. This is the setup. Looking at the actual results, you have larvae switched between urtica and salix. The control lines, log phone change of the control lines in the x-axis. On the y-axis, you have the switched larvae in both directions, from urtica to salix, from salix to urtica. And you have this for the 2 and the 17-hour sampling point. And you see after 2 hours, it's mostly expression that still corresponds to the first host, while after 17 hours, this changed a lot. There are not many differentially expressed genes, but some of them, or most of them, show stress response or something which is an adjustment to the new host. This overall low number of genes, differentially expressed genes here, could be because these hosts are relatively similar in their properties. So it could be that just very shared pathways are used to process these plants. Looking at larvae that were switched between salix and rebus, after 2 hours, the pattern looks the same. But after 17 hours, there's a bit of a difference. Yes, from rebus to salix, we see some adjustments, still a lot going on with host 1. But after 17 hours, we see that there are a lot of genes that are differentially expressed and quite some that show already an adjustment to the new host environment. And I said before, shared pathways, in this case, it's the opposite. Rebus has a very particular chemical component set. It, for instance, includes cyanogenic glucosides. So components that need rather specialized processes to be metabolized. And this is even clearer when we look at the comparison between, or the switch between urtica and rebus. 2 hours, as we expected. 17 hours, also as we expected, with adjustments to both new host environments, higher number of differentially expressed genes on rebus. And this is also an agreement to what we see on this bubble PCAs at the beginning, opposite gene expression patterns. Great. So we see there is flexibility in the transcription response, and we also see that the extent of the number of genes that are differentially expressed really depend on the direction and the specific host that is involved. And based on this, we assume that some hosts seem to be more challenging. And if something is more challenging, we assume that this must also be associated with some sort of physiological cost, and so we also checked whether the switch has an effect on the performance of the larva. On the performance of the larva, and this is why we calculated the growth rate as the log of the weight, pupil weight divided by the developmental time. And although we see these clear differences in the transcription patterns, the performance looked basically all the same. There was no significant difference. So it's not only a higher flexibility at gene expression level, but also at a physiological level, they can compensate very well for potential costs. Great. Looking back to our questions, we could show that Larvae can flexibly switch to a new host environment on a gene expression level, and we could also clearly see that there is a temporal component to it. So after 2 hours, it's host 1 specific gene expression. After 17 hours, we see clear adjustment to the new host. But, as I said before, the host plant itself and the direction of the switch matter, and some hosts seem to be more challenging. More genes are involved to regain the cellular homeostasis again. And one thing that I skipped because of time reasons, but I still wanted to mention it, we did not only check what are the categories of the different differentially expressed genes, but we also checked what are these genes actually doing. And we mostly found genes involved in metabolism, stress response, and signaling. So exactly what we predicted for the different categories. And we also compared this result to a previous study, and we saw quite some overlap, just confirming that these genes are definitely crucial for the user for the particular plant. Great. Overall summary conclusion, we could confirm host-specific gene expression. I don't know how many times I've said this term now. They are quite flexible in the transcriptional and physiological response, but why is this important? It makes a larvae quite resilient to changes in the environment. Let's say there's an oviposition mistake or the quality of the plant is decreasing. But maybe more importantly, in an evolutionary context, adaptations to one particular plant could also be used as pre-adaptations to a novel host. So it could help the colonization of new host plants, be involved in host shifts and so on. And although we did not see physiological trade-offs in this study, we cannot fully rule out that they never were involved in this entire evolution of a host plant repertoire. And to quote Mariana Braga, yes, larvae are parasites, so we can also assume that these dynamics that we see could also be helpful to understand other interspecific interactions. Right. With this, I want to thank all the people involved in this project. And you for listening. Thank you so much. And the last second, I want to just use for a short moment of bragging, because if you want to read more on this study, it's just got published in Molecular Ecology. Thank you.